Hello, everyone. Welcome. If you're seeing my face now again for the second week in a row, I apologize. I hope you had a fun brain hack. My name's Aki Nikoladis. I'm the chair of the Open Science SIG, and I'm super excited to have these wonderful guests, panelists today, speaking with us about some challenges in open science. I'll let them introduce themselves, and then we'll all just jump into a moderated discussion. Olivia, would you like to go first?
Yeah, thanks. My name's Olivia Guest. I'm a postdoc at the Donders in the Netherlands. My relationship to open science is kind of started maybe from learning about open source a very long time ago, and then that kind of turning into a sort of larger movement and larger understanding. I guess I do everything that has to do with open science, and also like noticing things like potentially like lacking inclusivity maybe, or lacking diversity in some areas. So maybe a bit more of an emphasis on that if I was to really put an emphasis on anything. But who should I just name someone else, or Danielle?
Hi, I'm Danielle. I am a second-year PhD student at the University of Surrey and partnered with Imperial College London. And my relationship to open science is a very loving one. We've been together for, I think about 2 years. When I first started my PhD, it's when I really got into open science. And it's been a steady love affair ever since. But my focus, the most perhaps fruitful part of my relationship with open science, is more in relationship to coding. So Aki, you said we can plug whenever we want. So this will come up often. My biggest experience with open science is through the Surrey Reproducibility Society. And through that, the coding workshops I organize as a part of the Surrey Reproducibility Society. So I'm quite interested in how coding can be used to further open science and coding practices that are best in line with open science, in addition to the myriad of other important open science facets. And I will pass it on to Mai-Lan.
Hi there, I'm Mai-Lan Ho. I am a pediatric and adult neuroradiologist. I direct radiology research and advanced neuroimaging and imaging genomics at Nationwide Children's Hospital and the Ohio State University in Columbus, Ohio. My relationship to open science is that I started out as an engineer, so I have a lot of interest in first principles and access to data and so forth. And particularly, I think in pediatrics, the issues with brain development, genetics, normal variants, pitfalls, all of that is very important in terms of being able to diagnose normal before you can actually figure out what the pathology is. And so I think particularly for pediatric data sets, unlike some of the adult human brain connectome and so forth, the baby brain connectome and adolescent stuff is somewhat lagging. And so I think understanding of that, physiology is very important for people to understand, as well as the variations in terms of MRI techniques that can affect how we analyze resting state, structural and functional connectivity.
All right, so I think that leads to me. So hi, I'm Raghavendra Selvan. You can call me Raghav for short, and that's perfectly fine. I'm an assistant professor at the University of Copenhagen. So I work in the machine learning section, computer science department. So my relationship with sort of open science, also like Danielle, started from software. So I've been part of the free software movement back in India during my student days for more than now, I think, I started in 2008, so long back. So I've been associated with that movement for long. And since then, that I've progressed into research and primarily doing machine learning for medical image analysis. I hold a lot of interest in having, the code and the data sets, available, which will for reproducibility and also just accessibility across geographic locations. I think that's kind of one of the important things right now, that I'm in Denmark and I've also been in India. So I see this sort of how to make it a bit more equitable. And then I think open science is a way I see it through. So yeah, that's about me.
Thank you, Raga. All right. Well, I think each of you kind of touch on topics that I kind of each offer a different facet to this idea of challenges in open science. So some of you have talked about, you know, issues of inclusivity. Others have talked about issues of accessibility, of knowing what norm is before we can sort of discuss what dysfunction looks like, et cetera. I'm kind of curious to hear what, if you guys could speak a little bit more about what you think are maybe the number one for you, or maybe in general, challenge that you think open science is facing, particularly in terms of adoption. So kind of moving from a perspective of open science being more of a fringe effort to being something that's more more common and you know, equivalent with what people would just call science.
Yeah, I can speak a little bit on that, if that's all right.
Yeah.
So thinking about, you know, how do we shift open science from this very popular grassroots movement to a more stable, steady, you know, structural change? I think what's important is to not keep the burden of the movement solely on the shoulders of early career researchers, but to get more established structural institutions such as funders, such as publishing bodies, such as universities and other institutions to wholeheartedly grasp the open science movement as well. And I think that open science has been an incredibly successful bottom-up movement, but in order to maintain the impact it's had, not just for this generation of incredibly passionate researchers, but to continue forward for all others, it needs to result in structural change. And that can only happen at the highest echelons of the structure of the research ecosystems we participate in.
That's a great point.
Yeah. I think continuing with what Danielle said, so I think sort of the elephant in this open science room is I think is infrastructure. So I think, we could, to a large extent, there's been a focus on open access, publishing or source code. And, so back during my free software days, we used to say, to run free software, you need hardware. So I think now when we do research, also we need a bit of a noise.
Some feedback. Maybe I'll mute myself. I guess if you don't want to talk, just go ahead and maybe mute. Maybe that'll make.
Yeah. So I think it's currently also a lot of sort of the resistance to open science also is due to constraints in resources. This could be infrastructure in terms of hardware or software licensing. And, I think, so there, what Danielle said, that if it comes from like a top-down structural change, for instance, the insistence in EU for open access publishing, where all the publishing agencies or the funding agencies have gotten together trying to persuade all research be put out in open access. I think that was a lot of pressure which came from the grassroots movement. I think we need to try and now get together and then see how we can enforce more of this, the top-down approach. So, okay, so I'm just going to mute myself just to see if it is me.
Yeah, I think from the perspective of radiologists, one thing that people don't realize is just how much technical variability and how much that affects post-processing and potentially scientific interpretation. So if you look at a CT scan, right, that's just based on density. The quality assurance between different CT scanners anywhere in the world is supposed to be 98%, right? So you can only have up to 2% difference in Hounsfield units in terms of tissue density. But guess for an MR, the average amount of variability, even just for a T1 sequence, not resting state functional MRI or DTI or anything else, but just a simple anatomic 3D T1 sequence. It's about 35%. So just imagine, you're sending this stuff through FreeSurfer, right, to look at, brain volumes and anatomic this and that. And even assuming that you have a structurally normal brain, that's the level of variation that you're seeing. Then you get into the whole, physiology of like cerebral blood flow with fMRIs and what kind of paradigm are you doing and all of this, and what coils do you have, what magnet strength, signal to noise. So it gets infinitely more hairy. And so there are two possible approaches, right? One is to try to harmonize protocols, which is only partially realistic because there's so many different MRI vendors available that you can try to do that, but still it's kind of unrealistic to expect everyone in the world to use exactly the same protocol. The other approach, of course, is deep learning, to try to use essentially unstructured, unsupervised data, but to have massive amounts of it so that you can try to, mathematically correct for a lot of this inherent variability, but that essentially requires an incredible amount of data sharing across institutions, which is also in itself controversial. And especially for smaller, more unique data sets like pediatrics or rare diseases, that becomes very challenging and scientifically hairy. So I think, collaboration is really key, and just being aware of how much technique can affect results is also really important. So there are websites like Neurosynth, right, that deal with some of this, massive grouping of data. A lot of that's more neuropsychological type research. But again, I think that idea of pooling data and trying to look very closely at the technical variations is very important.
Can I just ask a point of information? So you said it was 98% for a CAT scan, accurate for a T1 image, 35%?
35% is the degree of difference. So like 2% difference, in CT scans anywhere in the world, whereas around average 35 variation between different just T1 anatomic scans for MR. Yeah.
Yeah. Well, I guess that I'm glad that someone actually brought that up right off the bat. That a number on one scanner doesn't mean the same number on another scanner. That's an absolutely that's a bit of a non-starter in some ways to getting towards something like biomarkers of either cognition or psychiatric
correct
illness. Yeah. So okay, so how do we fix that? I mean, open science is definitely like a response, can be considered a response to that, right? We're going to share protocols, we're going to share analysis, we're going to share theory, etc. But I mean, speaking from the kind of responding to you guys discussing top-down change, and Mai-Lan, your point about vendors are probably not going to arrive at the same protocol, like. Is there any other hope of organized or top-down structure that can be used to, make some kind of more coherence between different, whether that's a post-processing thing where, like, all the Philips scanners get changed in this way and all the GE scanners or something like that. Obviously, then there's head coil differences, et cetera, et cetera, and then all the acquisition sequences are different.
Yeah, I mean, I think it depends on what you're looking for. So, like the Human Connecting Project, and now they have like Baby Brain and ABCD. So stuff, I'm sure they have the same things in Europe, but they're very, very controlled, right? So if you sign up for it, basically you're running your scanners whole time, just fixing the protocols and harmonizing them and having people go, both phantoms and actual people flying around the country to scan themselves on each scanner and do the post-processing correction. So it's very, very structured. And then that data gets published as part of open access for people to analyze. I think, as you mentioned, the really important aspect of imaging, imaging is longitudinal, it's non-invasive, but the real power is to link it to biomarkers. So the key is really going to be, what are these clinical gold standards that we have, psychiatric testing or other kind of like disease metrics and things. And remember, I mean, those are variable too, right? Depending on what neurologist you get or psychiatrist or whatever. So there's variation too, but at least you can link it to something clinical that's accepted as some kind of, standardized outcome. And so right now we can do radiomic analysis, or we can do deep learning, we can extract a bunch of numbers, but what do those really mean? So we have to at least have some level of correlation. And so when you look at single institution or multi-center studies where they are trying to do this, there's clearly correlation. The problem is, how does it generalize? How does it translate? Every place is going to have their own, metrics, thresholds for each kind of biomarker. And so there is certainly a challenge in, like, how does this really become translatable to clinical care? And that's, I think, the big challenge between, the kind of research that we do in radiology, which is kind of like individual patient decision-based, and a lot of the neuropsychology literature, which is much larger and more wide-reaching, but has some trouble getting into translation. And so that's really the core issue. I would say that overall, it depends on what you're looking at too. So if you're looking at DTI and advanced diffusion, you're looking at microstructure, that's going to be more reproducible than fMRI, right? Just because cerebral blood flow is always going to vary with things. And so test retest reliability of fMRI, even task-based clinical, paradigms, is unfortunately poor to moderate in a study done just last year in a meta-analysis. But if you're putting together functional with structural connectivity, if you're putting that together with some anatomic stuff, then you have a little bit more reliability. So I think the use of multimodal biomarkers per se, and the correlation with clinical data, and both these bottom-up and top-down approaches. Like clearly, we have to show feasibility kind of on the ground, but then there have to be these overarching programs like Human Connecting Project, where we're trying to provide a level of standardization as well, technically, in post-processing lines.
Yeah, so I think adding to that point, right? Basically, if we have all of the, or not all, but all published data available, and then the pipelines sort of also transparently published out there. So one could take up like a standardized streamlining approach where different pipelines could perhaps be, compared, and that is conditioned on the availability of the data and then the protocols that are being used. And I think in this regard, I would like to use the opportunity to plug so what we are doing. So I'm actually chairing the neuroimaging research pipelines work group at the Sustainability and Environment Action, SIG. And there, that's what we are trying to do. Basically, pull in, so we're starting out now, but our moonshot goal is to basically have like a framework where you can plug in a data set or a pipeline, and then we should be able to kind of, compare it or assess it, based on different criteria. And this is basically dependent on sort of a lot of open science expectations here, right? That the data is available and the code is available, and then, we all adhere to some open protocols. So I think this is where I think sort of the strong culture of making, not seeing science, like in neuroimaging and generally, as something different for, when you say open science, if that becomes sort of the practice, then I don't think we will have a lot of the problems, because I think one of the things is also to say that doing open science is more work. I think that perception should change. That should be sort of the de facto standard. And because it is so, when the science is not open, that's when it creates a lot more hurdles in reproducibility. And so I think we should change that perception. And I hope, with all our work, we will make some dent there.
But it is more work, right? I mean, there is a disadvantage to, there is some, you can argue that there's some disadvantages, right, to contributing to open source projects. You can have a closed source project, maybe that takes, 60% or 70% of the developer time that the open science equivalent of that would be. Obviously, the open science might have a bigger impact on the field in the long run, but over time, and this is going to get into hiring practices and those kinds of things. They're not really geared to evaluate people on their contributions to open projects.
I think Danielle, you had a point there.
And there's a lot of interesting comments in the chat too that I'm gonna be looping back into this conversation.
Yeah. Oh, you'll just see me type in loads. Yes, and you were mentioning that open science is perhaps a bit more expensive and difficult thing to do. And I say expensive, I mean both time and resource and energy intensive. I like to think more like Raghav's idea, where you shift your thinking of open science as a burden and more as an opportunity. And there are several really good papers that also, that support this line of thinking. I think Florian Markowitz's, 7 or 5 selfish reasons to work reproducibility sums that up quite nicely, and I'll try to find it and link it into the chat. But working in an open way, yes, it is often more time intensive, but you can make it worth your time within the current publisher perish atmosphere by publishing your methods, publishing your code. You can get credit in a system you dislike while still striving to make change for a system you prefer.
Yeah.
I don't know if this is maybe taking the interpretation of more or less work to a extreme, but I would almost argue that doing things openly could technically be less work, or at least different work or equal work, because in many ways, if people share successes and failures, like for example in computational modeling, if you share like your code base, in many ways the cumulative effect of people sharing their code base could ultimately mean that you do less work going forwards, because you don't have to write, for example, a library from scratch, or you don't have to go down a route that other people have already explored. So I mean, I think the idea that something is more work depends on exactly how you frame it, and not necessarily if it is more work in a kind of wider framework as a kind of field-wide more work. But I also think that in many ways, we shouldn't really, depends how we want to think about our own science, but we shouldn't really be thinking about science as like an individualist endeavor, because that's not really how science kind of works. Like most of the kind of research that we do is probably going to be thrown away by future generations, and that's maybe completely fine, and that's maybe exactly how normal, healthy science is. So in many ways, doing it openly has these kind of historical benefits too for future historians, philosophers, and whatever sociologists of science who get to look at all this stuff more easily. And it does actually improve the understanding of our own science if we do it openly. So it allows like meta-scientific research to be carried out more easily, and so on. So I mean, I think maybe it's like a kind of a sneaky rhetorical point that seeps into the way that we think about our science when we think of open science as being more work. But I do also think, there is the other side, which is that we are putting publicly funded research quite often into private companies' hands when we do open science as well, unfortunately. GitHub is a private company, for example, owned by Microsoft, in fact. So I think there's a lot of points to make on all sides. And maybe I'm being super pedantic, but if I understand correctly, because I'm not exactly in the same area as other people, and I'm sure each person has their own unique perspective, but are people saying that the biggest barrier to open science is just people don't do enough open science?
I don't think that. I think that there's reasons why people don't do enough open science, and unfortunately, that I think it does go back to the individual. I mean, I agree with you. It's nicer to think about things in a communitarian sort of way, and I definitely think that way. I mean, science as a whole is like this great socialist experiment on like, can we share things and benefit each other or something like that? But at the end of the day, like, people leaving science has to do with, like, open scientists leave science all the time, and that has a lot to do with these kinds of individualistic decisions and sacrifices, and just, can they get a job? Can they get a job where their partner wants to get a job? Great points all around. Let me ask. The chat is super interested in what you guys are saying, so I just want to point out one of the questions. So Melanie Gatz says, "Dear Mai-Lan Ho, could you comment on multi-center studies such as the Enigma studies? They're not as aligned as ABCD, but then they do align their processing somewhat as well with QC." So let's just get that, and then we have some more stuff to talk about.
Yeah, I think so. I do a lot of neurogenetics, and what I like about Enigma is that they're trying to link imaging to genomic information. I would just, because I deal a lot with genomicists, I would provide the caveat that there's even more variability and more potentially junk data when you're dealing with the whole genome than in imaging, where at least you can draw like a region of interest or volume of interest around what you're thinking, or like strip the skull or whatnot. So I think it's a very good initiative. And they do have, I know, different working groups for different types of disorders. So obviously, like epilepsy or brain tumor, where you actually have a lesion on the scan and you can't just use FreeSurfer in its native state, is very different from like ADHD or autism or something type research. So I agree that those are all different, working groups, and they need to have different skill sets. I think not having as structured or streamlined acquisition and post-processing has pluses and minuses. So I do think when you're dealing with real world data sets, there's no way you can harmonize everything. And so it makes sense to have, reasonably aligned, but not exactly the same, because it will never be exact. The caveat there is that when you're doing the processing, you cannot rely on traditional, you cannot rely on traditional mathematical operations or the traditional types of processing software that you would normally utilize for, one center or like a few center neuroimage processing. You're going to have to use some kind of a deep learning big data approach, because now you have factors that you don't realize, right? You have things like magnetic field and different parameters and stuff like that. And so deep learning is unfortunately a little bit black box, but you're going to have to utilize like an approach to correct for some of the inherent bias in these data sets. So I very much support more diversified data sets. And you also have to make sure that your training set doesn't have inherent bias. But the more that you have this diversification data, the more you're going to rely on big data approaches to be able to analyze them in a meaningful way. Even this goes even more for genomic data sets, because there's very little, right? There's very little like certain exome and certain, genetics is not gold standard, right? So I deal with this a lot with the rare diseases. So when you do a whole exome, which is like the clinical whole exome. So in clinical practice, if someone has a disorder, suspected disorder, you can do a single gene test, which is like slam dunk, but that's very rare these days, right? Because there's so many different molecular pathways with overlap. So you can do a gene panel. So if you have like, let's say epilepsy, or developmental delay, right? Hearing loss. So there will be a panel of like however many, let's say like hundreds of genes or whatever that are currently reported and known to be linked. You can still miss stuff. So that can reflex into a whole exome, which is where you're looking at all like the coding genes. But even there, right, so if you have a somatic mutation, if it's not a germline mutation that you inherited from your parents, but it's something that you developed later on, it could affect only a part of your body. So it might be only in the tissue of the, let's say, the brain tumor or the brain lesion. Or you might be able to detect it in the blood, but at a very, very much lower concentration. So unless you're doing a deep sequencing, so you're talking like millions, hundreds of millions, billions of times for that one gene, you might miss that mutation. So the more that you can actually phenotype using brain MRI, and give yourself like a pretest probability to say like, these are the genes I'm looking for, the more they can focus on that. So we do it all the time. We miss meaningful variants all the time, either because we didn't sequence deep enough or because the gene wasn't known to be of significance then, and it was only like a couple years later with the case report here and there that you start to discover it. So genetics is also not a gold standard, and you also have to get the right area of tissue. But I do think that those two large data sets are very, very useful to be linking together. It's just that we have this whole problem of that, unless you're dealing with a very rare disorder that has like a single gene link to it, the vastness of the genome and the degree of information we get, even just from evolutionary biology over like whole Homo sapiens career, it's not enough for deep learning methodologies by themselves to apply. So you need to have like an informed combination of more targeted machine learning type approaches and the deep learning. So it's kind of with this combination of handcrafted extracted metrics and big data approaches, which is exactly the same like radiology.
Thank you. That's super interesting. You mentioned this one thing that I kind of want to touch on about how you have to make sure that there's not bias in your data sets.
Correct.
And I think like all of us can sort of discuss the issue of bias, not only in our data collection, but also in who's doing the data collection. And, one of the, at least in open science, I'm not going to say this as science as a whole, but open science kind of as a subculture of science tends to be predominantly biased towards, whiteness, maleness, Europeanness, North Americanness, etc. I'm kind of like seeing that, we're talking a lot about the technical elements and technical challenges of, open science, which are substantial and obviously like we need to address. I'm kind of wondering if we could maybe shift gears a little bit, talk a little bit about the sort of social, cultural limitations of open science, right? Because, after all, what we're talking about is changing a culture, right? It's not like, oh, you weren't running the right Jupyter notebook, or you didn't have like, oh, you had Python installed, or you had MATLAB installed. Now you need to have Python installed, and that's going to fix everything, right? It's like transition to open science is a cultural one, right? So I'm kind of curious if I could get some feedback on this sort of turn. What do you think? Is the technical burden actually the one that is the more important one, or do you think the social one is the more important one, et cetera? I'm not going to take a point on it. I just want to hear what you think.
Oh, I'd like to contribute to that. Just, kind of talking about the intersection of, the technical aspect of open science, the social aspect, and how it relates to open science as a culture, and hopefully having it become more of a norm rather than a, what I refer to as a culture. Because I often, I hear people often refer to kind of people like me who are very enthusiastic about open science as being somewhat part of a cult. And I like to say to them that, well, actually, you practice open science in this way if I know them well enough, and I can point out either they, share their data, share their code, and then I often invite them to watch Paywall the movie with me, which if you haven't, it's great. I'll put it in. But shifting the social part away from being viewed as an other thing and more as everyone does open science to a certain degree, and we can all try to make open science more from a higher degree, more of the norm. And I think what's really cool is that, the technical challenges to open science, I think, are maybe some of the more initial barriers. But for me, I wanted to get better at research, so I joined more open science things to learn more about how I can improve my research technique. But the reason I stay in open science is actually because of the community. Everyone's really lovely and helpful and willing to help me get better at these challenging techniques. And so I don't know if this is an experience shared by you guys, but perhaps, we can lure people in with the promise of better research practice, but keep people in with this, very open, kind, and supportive community that open science has fostered.
So I think I have sort of a bit more, perhaps, like a high-level structural point about science in general, and then so I think you were pointing at how it's predominantly like global north that is driving much of the research and science, and also open science. And I think there's no shortcut to solving this problem other than including everybody, right? So how do we make it more accessible across the globe? And that is not an easy thing to do, except that, I don't know, that these are like really big problems. But of course there are, I'm thinking, why aren't there any sort of international funding consortia of some sort, so where you don't stop at the borders of national borders, for instance. So we need to, there's no shortcut to this, in my opinion, at all. Of course, we can have some of these simple, technical things that we could do to kind of encourage this behavior, but participatory science is when it will be diversified. So, we cannot have like a data set that is truly unbiased if we have like a majority of the population not participating in that science. So I kind of strongly believe in that. And how do we solve it? I mean, yeah, like I said, we should have like a big structural change where we can end up with like a consortium which will fund, projects across the globe, and we can have crisscrossing of. I'm thinking in my head, it's like a research NATO, So why not? So yeah, that's
that's
sort of
so
like,
cool.
All right.
So my take on this, obviously, I take the previous points too, but I think something that happens very often in these discussions that I find extremely disturbing is the complete erasure of South America and Latin America, who basically have been complete pioneers of open science. They've been doing it before all of us, and they do like the bulk of it as well. So the Global South in general actually does tons of open science, and we know this. And yet ignore it for some reason in a lot of these discussions. So I think part of the problem of diversity in open science is that we erase all the work that the Global South is actually already doing. And they do it in many ways that we might not even be aware of. So the erasure is implicit, but they're also explicitly doing things that we're all aware of, and we still decide to erase them, which I think is very shameful and shocking, and is something we should all strive to stop, because it's a kind of false narrative to say, like, oh, we need to reach out to these people and teach them how to do open science when they've been doing it since before we've been doing it. So that's one point. The second point that ties into that I'd like to highlight is that these people are also driven by slightly different reasons. So like colleagues of mine who are in the global south, quite often they're driven to use, for example, R instead of SPSS, not for the same reasons, not because it's cool, not because it's open source. They don't necessarily care about that, but because it's free, and it pushes them to learn these, you know, in some senses more complex tools. Like you could say, I don't know, whatever, you could say R is more complex than SPSS perhaps. So they're pushed to learn these tools because they essentially can't afford to buy a license for SPSS or a license for MATLAB or whatever, right? So they're pushed to learn whatever, to use LaTeX and not Word, because Word is not free. So I think it's important to remember that the global south is doing open science, and it's our job to credit them with the stuff that they do. I don't know, I feel like it's a bit of a moot point to say, like, we should, I don't know, like, reach out and fund them. Maybe what we should do, of course we should reach out and fund them, but maybe what we should do is, like, reach out and actually talk to them. They'll probably teach us more things than we can teach them.
Yeah. So I totally agree with you. So my point wasn't about open science in general, but then just science as a barrier. The first point that I mentioned was in terms of infrastructure barriers. And I think that is something which comes sort of like a lot of personal experience also. Like I said, it's so in terms of access to publications, to conference fees, to travel expenses. And I'm saying, so this is like so the whole environment is somehow thought out, and the whole, like, even the OHBM registration fees and things like that, right? So, I mean, that's the point. It wasn't at all about open science, because, like I said, I've been in the Free Software Foundation, for instance, like 10 years back in India. We did that because we couldn't afford Microsoft, license. Linux was cool because of that. But I mean,
I wasn't specifically disagreeing with you, exactly. By the way. I was saying like the broader kind of framing,
but I recognize that, because the whole framing of this whole debate is also done in, not our debate, I mean, but then generally around this is also like that. But yeah, very great points there. Cool.
So my commentary on culture shift and barriers is going to be about data privacy. So this is like a big thing in especially with the clinical research. And obviously, a lot of these neuropsych studies are more like college students and things, healthy adults who are coming in and signing the IRB, so it's a little bit easier. But there's an incredible amount of controversy around privacy. So in the US, it's called HIPAA. And so actually utilizing the data, if you utilize it for, let's say, an institutional research project, you would IRB approval. But if you want to do it for AI, and potentially if you want to do multi-center AI or any kind of, data sharing, that becomes much, much more hairy. And so there are a couple approaches to this. One is just to actually approach each individual, either prospectively or retrospectively, and say, do you want your data to be part of an AI project? And you know what the answer is going to be most of the time. It's also logistically very unrealistic. The other thing that some hospitals are doing are kind of like an inherent consent, which I also don't really, that's also very ethically gray, that they're just saying, well, when you come to this hospital, you're basically automatically assuming that some of your stuff will be used for this and that, and we don't exactly know what it'll be, but you're inherently consenting. And that's a problem too. And so I don't know what the answer is. There are some things like distributed or federated AI where, each institution can keep onto their own protected health information, but the AI bot can essentially go in and optimize the algorithm for each of them without the centers sharing with each other. And obviously data is the prize now, right? That no one wants to just provide all of this hard-earned, curated data out to the world when they spent so much time, working on it. And I know that's kind of like not in the spirit of open science, but unfortunately, a lot of these are kind of although they're nonprofit institutions, there is a level of like, okay, there's a bottom line here. So especially when you're dealing with disease processes, rare diseases, or highly curated things, it's going to be hard to convince people to be completely open, but at least some of these like distributed AI models. One thing that's interesting is the difference in healthcare systems between, let's say, North America, South America, and Europe, for instance. So I've done a few projects with people in Europe, and for instance, Karolinska, right? They asked me to look at the brain MRIs for a bunch of twins with different autism spectrum disorders, and I said, well, do we have to like look at all the? Do we have to collaborate with all the other institutions in Switzerland? They're like, no, actually, every single set of twins or triplets that's born in the country goes to Karolinska and gets sequenced. I'm like, God, that's amazing. So they have the same thing in Europe, right? Where, or they used to anyway, where for different neurogenetic disorders, like Mario van der Knaap, who I'm sure a lot of you recognize the name, but she's an expert neurologist in all white matter disorders. So no matter where they are in Europe, you know, she could get the brain MRI, she could get the sequencing. And so she became an expert in that field. But now they've passed new privacy laws, and that's now restricting the research over there as well. And so again, I mean, it's kind of like, you know, there's kind of the patient's privacy rights, the individual's rights, and there's the kind of, you know, community and global science. And so there's a tension there. I'm not saying that there is a right answer, but clearly there is that tension between the individual and the you know, overall scientific, you know, forwarding science. So
yeah. Well, thank you all for your comments, for your insights today. I'm super appreciative that we were able to get you all for this short panel. Unfortunately, we're running already late, so I'm gonna just jump off and wrap up. Thank you everyone for listening in, and we'll see you at the rest of the conference.
Thank you guys. I'll
thank
see
you so much.
Sorry everybody, sorry for the delay. We are trying to add everybody and then we could start. I'm sorry for the delay. So we have 30 minutes to go over this, and 25 minutes for the panel, then 5 minutes for question.
Is that better?
Yeah, much better. Hi.
Can you guys hear me?
Great, finally,
Cool.
We're all here. Thank you very much, everyone, for bearing with us while they brought us up on stage. So I'm Charlotte Ray from the University of Sussex in the UK. I'm the chair of the new OHBM Sustainability and Environment Action Special Interest Group. We were founded last year with the aim of tackling the environmental footprint of our academic research activities, from human brain scanning research, of course, including open neuroimaging, through to the cost of the annual meeting. And thinking specifically about open neuroimaging, we have a panel here today of 4 colleagues who've been thinking about how to ensure our open neuroimaging workflows are as sustainable as possible. So welcome to Nikhil, Dustin, Young Guy, and Winson, all of whom are members of our special interest group. I'll let them introduce themselves in a moment, and I just want to give a reminder of why this is so important. We're all scientists, so I think you all know how stuffed we are if we don't decarbonise the whole of society and stop the mass biodiversity loss we're causing. And for more on that, come to our symposium. So I want to open with a question around which aspects of open neuroimaging practices are already sustainable and we should be promoting, and which aspects are not so sustainable and why. And which of those are the biggest issues and how could we address those? I'll come to Nikhil first.
Hi, can you guys hear me? Cool. So yeah, just briefly, I'm a postdoc at the MNI in McGill. I've been working on the intersection of computational neuroimaging pipelines and deep learning and reproducibility for the last few years. And having worked in a few different labs with different scales of data and computational resources, I have been kind of worried about the sustainability for the last few months or maybe a little bit longer, and I've been working on it. So to answer this question, I think the brief answer is we don't know which ones are sustainable. I think there needs to be a lot of benchmarking needs to be done. In terms of at the individual level or individual researcher level, as well as infrastructural level, what is our carbon footprint and what is the carbon emissions that come from data storage, as well as the computational resources. But few positive things are the overlap between the last panel we had, the open science practices or the FAIR data standards or reproducibility in general. These type of practices are definitely helpful in terms of maintaining sustainable workflows and infrastructure. At the same time, I think there is a financial incentive for many data centers, especially the commercial ones that are making the data centers and the compute cloud structures more efficient as possible. And there's also a push for renewable energy sources that is going on. So those things are encouraging, but like I said, the short answer is we don't know what the impact of our research is at this moment. Nope, that's it for me.
Thanks. Shall we come to Dustin next?
Sure. I'm Dustin Murchevsky. I'm a data scientist on the data science and sharing team at the National Institute of Mental Health. And so I want to disagree a little bit with Nikhil in that we do know a little bit. And one of the things that is sustainable is not using resources. And so something that that I want to say that takes inspiration from the open science movement is being as thoughtful as you can about what you're going to do before you do it in terms of using resources. So that could be, if you're designing a study, trying to think about, does data already exist that can address my research question? So data reuse. Then you don't need to use the resources to collect that data. Or if you're going to put together a large processing job on your HPC with HPC or something like that, or HCP, Human Connectome Project, maybe do some investigation to see if those derivatives exist somewhere else, so that you can reuse that data. Or you could share data sets at your institution so that you don't have to download the entire UK Biobank data set for each lab, assuming that the data use agreement is okay with that. Or it could be, if you're ready to send a job to your HPC, then do some benchmarking on one subject or 5 subjects to make sure that you know what the result of that processing is going to be, so that you don't have to run an entire pipeline on a massive data set and then, in hindsight, have to rerun it.
Thank you. Let's come to Young Guy.
Hi, my name is Young Jae. I'm in New York. I'm a postdoc at Mount Sinai Hospital. I do a little bit of neuroimaging, but I do a lot of genetics and genomic analysis, investigating Alzheimer's disease and other human diseases. So I come from a little bit of a different background from traditional neuroscientists or neuroimaging scientists. I think I concur with Nikhil. Personally, I don't think we know much. Personally, I don't think we know what we need to do. There are some things that we do know in general sense what we should do, but I think there are some things that we're doing well, which I think is data sharing, which I have also benefited from. So, instance like Loni has lots of Alzheimer's disease data available there. So I was able to use that data, and UK Biobank also has lots of MRI data there. So being able to utilize those data, I think that's the field of neuroimaging is getting better at, but I'm sure it's not perfect, but that's one way to make this research more sustainable. But at large, at more individual level, we don't know what we need to do, I think. So what we need to do as a team is figuring out priority, what we need to prioritize, what we need to figure out for the community to get involved. So I think 3 of us are part of a carbon footprint team, and we're trying to figure out what we need to do. And we have some small goals that we are targeting. So I think we will learn what we need to do soon.
Thanks, young guy. And on to Winson.
Hi, I'm Winson. I'm a third-year PhD student at Texas Tech University. So just a brief introduction, I work mainly with on meditation data sets, looking at meditation, how it affects the brain and behavior. I co with Youngjae and Nikhil. So I think we don't really know the impact of our practices in neuroimaging practices and what those we think are sustainable, what their impact are, because I think it's relatively new. But also, like echo Youngjae, that we have platforms like repositories, we have sharing platforms and all that. But I think I'm coming from the structural point of view about what we should or we could promote is coming together as an organization to tackle sustainable neuroimaging rather than individual. Because as an individual, you're just one person. You don't have an organization backing you up. You don't have much power in the sense of influencing other people because you're just one person. So I guess coming together as a lab, probably department, university, or joining CSIC will be one of your options, because we all are how do I say, we are concerned about the environment and the impact of what we do on other on the society as well. So I think that is one of the important things we could promote to come together as an organization. And also I think what we could do is to include people from other disciplines like climate scientists, physicists, engineers who can provide or discuss with us on creating new, wouldn't say resources, but alternative resources or alternative methods for neuroimaging. Like, because we know that helium is non-renewable gas, so we could form a team to discuss what research could be done to look at renewable resources, what pipelines is effective, and how that affects the carbon footprint of our research as well.
Yeah, absolutely. So I think a lot of what you guys have picked up on is the complexity that's currently facing us, where there are some known unknowns and there are also some unknown unknowns for us. And I think a lot of what you've said, Dustin, is flagging that although we've got a lot of unknowns at the moment, we've got a handful of things that we think are knowns, or that, you know, we probably don't even need the hard data on exactly what the carbon footprint is to know that this would make a difference. And I think we can think of these as the kind of, you know, low-hanging fruits that are probably about optimizing your workflows, optimizing productivity, saving time and resources, which, you know, is the title of this panel, you know, around, like you were saying, if you're going to submit a big job to your cluster, just try it on a handful first, check out the outputs, and optimize that workflow. If you're downloading data from the HCP or the biobank, make sure you've just got the one institutional download. Clean up your files regularly. And these are all good habits for all of us as scientists anyway, that also probably help us create good open science resources to pass on to others. But I think what the three others said highlighted that we need to start filling in the knowledge gaps around the unknowns. And that's where our SIG is trying to come in and help fill those knowledge gaps. And part of this is just to quantify what's the nature of the problem, how big are the problems at different aspects along the pipeline. And we kind of need to empower ourselves with that knowledge before we can design resources that, Winson, you kind of pointed to a bit. And I kind of see that as a key role for us in the SIG of it being an international team. As you said, Winson, we can't expect individuals to go this alone. We've got to kind of produce community resources to help guide on this. So I've got a second question for you guys, which is almost a little bit philosophical, but thinking about open workflows and how we carry data all the way through. A lot of us are concerned with reproducible workflows. In your opinion, are fully reproducible workflows sustainable workflows. What might the issues be there, do you think? Should we go in the same order as before? Nikhil, Dustin, young guy, Winson.
Yes, I mean, really interesting question. And just going back to Dustin's point, I guess, yeah, there are some things we know intuitively what should be done in general. But like I said, like Charlotte said in her talk, those who have seen before, there are multiple levels at which we need to think this problem. And especially at infrastructural levels, there is not enough transparency in terms of how much energy the data centers are using or how efficient they are. And that needs to be benchmarked much more these days. For reproducibility, I think there's a common, it's going to be a bit of a trade-off, because to make things reproducible, we're going to have to replicate and rerun certain things, which is going to increase the computational burden. But I guess the underlying fact is, if the research is not reproducible, it's not sustainable. So there is no point if we don't know if our research is reproducible, then it cannot be sustainable in long term. So yeah, again, this goes back to my point where I have tried to work on this for the last few months in a concrete way, and it is rather tricky to quantify all the different types of permutations that can go into all the reproducible workflows, all the multiverse of reproducible workflows. It is challenging to quantify concretely. But again, more tools we build for improving reproducibility, more efficient things will be. So there's a trade-off more for sure in terms of we need to run more things to make to basically verify things are reproducible. But I think the payoff or the benefits of doing that are massive in terms of believing more into our science practices, and also reusing the stuff we already have, so we don't have to duplicate our effort in long term because it did not work or did not reproduce going back. So those are my initial thoughts on this.
Yeah, I agree with a lot of those comments. What I was going to say is something that I think Chris has already said in the chat. So thank you, Chris. He said, I would say that a fully reproducible workflow is effectively something that can be memonized. And somebody then said something. You can choose whether recomputation or retrieval is cheaper based on your conditions. So I was going to answer this question by saying, I mean, obviously with most things, it really depends. What are you reproducing? What are you reproducing here? Now, ideally, as Nikhil said, a fully reproducible pipeline is ideal, but it really depends on what the scientist is wanting to reproduce here. So if a scientist puts their project online, they've got the raw data, they've got all of the derivatives, they've got the code, and they've got the results. That's great. Then somebody can download all of that and reproduce it. That is still using resources to reproduce, and so reproducibility isn't inherently sustainable. Just like a comment that I wanted to make about data reuse, it's not inherently sustainable because people can just keep downloading the data over and over and over again. So you need to be thoughtful about how you're reusing it. So we have obligations about our personal behaviors about how the reproducibility and the reuse is going to be sustainable. But then it also depends, because data transfer uses resources, and you have to think about, now these are getting into the unknown knowns right now, about what resources are storing the data, what are the energy uses of the data centers, the sources. So if the data are stored on a data center that is completely sustainable, then that is a whole lot better. But then doing that data transfer might not be using power that is sustainable. So it really depends here on what the purposes are. But I think that irreproducible pipelines are not sustainable. I think we can probably say that.
Yeah, I agree. Non-reproducible pipeline is not only affecting sustainability, but it's a huge issue scientifically. So having a reproducible science, I think that should be a goal for many reasons, not just for sustainability issues. So not having a reproducible pipeline, having a valid finding that doesn't need validation from another cohort, study, cluster, computing power is definitely sustainable. So I do emphasize reproducibility. However, from last panel, there was someone who said T1 scan has a difference of 35%. So knowing that, I think that's a big issue in my opinion. I looked at longitudinal data for some of the data sets and it wasn't that big. I looked at variance, it wasn't that big to me. So I think the field needs to come into more consensus about how valid the data capture is, how consistent it is, and how reproducible it is. And us, we can work more on reproducible pipeline, having documentations and having more reproducible. But I think we're talking more very microscopic level. I think sustainability level is more macroscopic. So I definitely resonate with Greg has mentioned about optimizing, rather than optimizing workflow, thinking about select resources that are greener energy. So I was going to talk about this too. There are some tips that we can know and we can try, we can think about during our analysis. Like in Canada, it's more reproducible. Canada, it's more renewable. They're using hydropower, so it's more renewable energy. So using the cluster or computing core there might be more sustainable than using computing cluster, for example, in New York, which using some hydropower, but it's mixed with the fossil fuel. So thinking about the region and time of your analysis is useful. So for instance, let's say in winter in Canada, and the water's frozen, hydropower is not running so well. So maybe running a big analysis in Canada in winter might not be a good idea because they have to use fossil fuel to generate energy for that. But in summer in Canada, I think it's very excellent. The water is free flowing, hydropower is abundant. So the energy that you're using, the grid that is using the energy is very renewable. So thinking about these issues, so another way to think about is the time of using it. So for instance, during the day, a lot of non-scientists are using the electricity. So the electricity that is running the cluster might be replenished by fossil fuel. But if you run your analysis at night, then people are sleeping, they're not using a lot of electricity. So the energy that you're using is likely to be more renewable. So thinking about those things, timing, season, region, I think that's something that we can think about and we can implement right now without knowing how much impact we're making yet. But we do know we're making some kind of impact. So that's something to think about, I think.
Okay, so I'm going to go from also same more structural point of view. I echo with everyone. So with regards to the pipeline, we do not know how much variability, like what previous speaker in the previous session say T1 35%, that's a lot. And I think one thing we might want to think about is like what Dustin said, what we are doing with the data that with the pipeline. What's the intention behind that? If we're just trying to reproduce it, okay, that's good because we're trying to see if that data is reproducible or whether we could use that data. But I think on a deeper level, it comes back to the idea of falsifying the null hypothesis in science, and it comes back to the idea of whether the data will make sense. If it if it's not reproducible, it's not just sustainable, but it also says a lot about the theory behind the analysis behind, the thought behind, and the research question behind maybe how we carry on the research. And it could also inform a lot about what other researchers will look for, should be cautioned about when we are thinking about the entire process of research. Also, I think it also has to do with what the data actually means. If it's reproducible, does it mean that the science is reproducible, that the theory holds for now, or whether it's more than just that? I think we are not really sure. I think at this point, not really sure what reproducible data means, whether it's it stands for sustainability, whether it stands for science, whether it stands for research or other aspects. I think that is one thing that we should think about more, and how to tackle that, how to interpret those data and reproduce results, or results that are non-reproducible.
Yeah, so I think you've brought up there, Winson, the kind of even amongst ourselves, we don't necessarily know what does a reproducible workflow look like. And a staunch open science advocate might say you need to upload all the raw data, or to reproduce every single segment. Whereas someone like me, and maybe Dustin, you fall into this camp as well, we might say, well, can you just upload the pre-processed data? Is that enough? If we're talking about HCP stuff, or things that have been acquired on standard protocols, leaving the 35% variance in T1s aside. So that maybe that's another practical way that we can reduce the footprint of having all these files on servers that maybe aren't required. And Yonggai, you've given us some really practical tips. But I think something that the last round of questions just really picked up on was, even amongst ourselves who are really into this issue, there's quite a lot of variance in where we're kind of setting the bar for what cost we're willing to pay environmentally versus compute, essentially. So I think I'll just try and wrap up with the last round of questions around how do we draw that bar, that boundary? Even amongst ourselves, it's going to vary slightly. In the SIG, we're going to have to come up with some sort of heuristic to put out there. And we're the motivated ones, right? And everything we've just described puts quite a lot of burden on individuals to go away and make sure that they're running their scripts in an efficient way at certain times of day, accessing certain compute resources, if that's available even to them, for that have high renewables on them. So what about all those other people who aren't on this, crowdcast with us, who are not necessarily as concerned as us? How do we motivate them if they're going to be drawing the line somewhere else? So Nikhil, on to you first.
Motivating people is probably the hardest problem for a lot of things. From open science to reproducibility. I think motivating people to do the, supposedly right thing is a hard thing. But I think environmental concerns are pretty global. So from that perspective, I think as a scientist, we all are concerned in terms of our footprint and impact on the environment. For people who don't have access to more renewable sources and renewable data centers, I think that's a very difficult question, because based on at least stuff that I have been trying to look at, those are infrastructural issues. And so those infrastructural issues need to be dealt at organizational or governmental level. But again, more awareness we have, more we can put pressure on people or our institutes to sign up for green energy sources. And it's just like open science. More scientists believe in these things, we can push people or push institutions to make the right changes, infrastructural changes. Also, I think this is something I think Charlotte has mentioned before as well. I think thinking globally, I think despite where you are, I think slowing down the pace of our science and the whole this publish and perish type of race, I think if we slow this down, we will put less burden on the resources that we have. It may be data or compute themselves. So just slowing down things probably would be beneficial, not just for sustainability, but also reproducibility and open science. And just giving people more time to think about these issues apart from the pressures to publish or pressures to finish your analysis as soon as possible. But motivation is a very difficult question, I think.
If reducing our impact on climate change is not motivating enough, then I guess we have to gamify things. So everybody likes badges, everybody likes scores. And so, you know, one of the goals of our SIG is to quantify the carbon footprint of different studies. And so if we can put that number out there, and you can include that, let's say, in the at the end of each paper, along with acknowledgements and conflict of interest, you have a carbon footprint of your Earth, and that we can try to start gamifying it so that there's motivation.
Sorry, just to add to Dustin's point, I think he's exactly right. And there are tools, so which I forget to mention, there are tools already available to quantify the impact, environmental impact or carbon footprint of your computational pipelines. And we can share those here as well. Yeah, so there is, putting those in your papers, just like other metrics like accuracy or any other metrics would be really, really great in terms of motivating people or raising awareness of people of this issue.
So yeah, I mean we're on the same team. So Nikhil is developing the tools to quantify carbon footprint in some of the computational cores, and Dustin's working on figuring out infrastructure level how much carbon footprint we might be leaving from using the core. So we are all working on these generating ways to doing things to identify statistics on how much impact we're doing in terms of how much carbon footprint we're leaving. But definitely we need a lot of support from people who can increase awareness, people who can communicate with the big PIs, industry level, and also organization heads, because we can do a lot of things, we can try a lot of things, but definitely we will be needing help from them and convincing them that this is needed. We are doing research to for the better future, but you know, if we're not going to have a future due to sustainability issues, what's the point? So we need to convince someone at the overhead to help us.
I think we can also think about a think tank and writing a new paper regarding these heuristics and what people could do. I think there's already one, I think in 2019 or 2020. But I think that creating a think tank and the outcome of it is a new paper that talks about what neuroscientists could do and what are the potential outcomes, like reduce carbon footprints, like including what everyone said here about the statistics to churn out the carbon footprints, also for journals to include these, and incentives for those who, like gamifying, like what Dustin said. I think that might push people to be more aware of what the outcomes are and how that impacts the entire scientific community and also people living on Earth, because we're all living on this Earth. And I think resources are scarce and getting more scarce on these days. So I think that's pretty much it that we could do.
Great, thank you all. So I think we'll wrap up there. Just to reflect briefly on what Adina said in the chat, she's saying, you know, the programming language you use can, you know, even have an effect. So, you know, there are just so many considerations all along the pipeline, some of what we're already doing will have, you know, a net gain for the environment, some of which will have a net cost. And I think everything we've said today shows us that we already know a little bit about some of this, those low-hanging fruit we talked about, and Nikhil's just put some really useful resources in the chat. Do check those out for how we, you know, can start to measure these things. But we've really got a lot of work ahead of us in the SIG to really pin this down. And then Winson, you know, as you pointed towards, we are looking to put out academic papers on this, you know, perhaps via something like Aperture, the OHBM journal. We're looking to put out resources, we're looking to put out code. But of course, we need everyone in the community to get on board with this, you know, approach. It's got to be the only way that we do open neuroimaging going forwards, because as all the other panelists have said, if we don't change how we're doing things, we really are stuffed within our lifetimes and those of our trainees. And do come to our symposium to hear more about just how bad things are and how important it is that we all crack on with solving these issues. So thank you very much to the four panelists. It's been wonderful to have you. Many thanks indeed to all of you who have attended. I hope we've inspired you to start thinking about these issues and think about how you could incorporate some of this into your own workflows and practices. And a very good morning, day or night, wherever you are. And we look forward to seeing you at a CSIG event in the meeting platform over the week. Goodbye.